just as a brief introduction, as everybody knows here, that acute liver failure involves an initial liver insult caused by several chemical compounds, including acetaminophen, or APAP as we will call later on, which results in sudden and massive cell death, leading to release of damage-associated molecular patterns such as genomic DNA, HMPG1, causing an inflammatory response, which besides perpetuating the initial insults, can result with time, if doesn't resolve, with multi-organ failure. So on this side, you can see that recently in the literature, the microRNAs, which we learned yesterday in the basic science workshop, are short non-coding RNA molecules which regulate gene expression, have been recently in the spotlight about liver injury, their involvement in liver injury. Basically here this cartoon shows that after the liver gets injured, microRNAs can be released into the bloodstream in different forms, can be bound to proteins or packed into exosome, microvesicles, apoptotic bodies, and then can function as either short-range communication, cell-to-cell in the liver, or as a distant communication molecules, liver to other organs. So recently our group has characterized a novel porcine model for APAP-induced acute liver failure, and this model has been been proven to be clinically relevant, as it allows monitoring clinical markers of disease progression as they evolve from induction of acute liver failure through acute liver injury, acute liver failure, and death. And here it shows how the clinical parameters, international normalized ratio, creatinine, and CCP markers for liver, kidney, and brain injury respectively evolve with the progression of the disease. So the rationale and the questions for this project were basically three. The first one is there any change in the plasma levels of both total and tissue-specific liver, kidney, and brain microRNAs during the progression of acute liver injury through acute liver failure and to death? And if so, could be these microRNAs used as a biomarkers of specific tissue injury in the APAP-induced acute liver failure pig model? And lastly, we wanted to see if the increase in these microRNA levels could help us visualize the timeline of organ dysfunction and injury in acute liver failure. So experimentally we had two groups of animals. We had 3 control sham-operated animals and 6 acute liver failure pigs. And this cartoon shows the timeline of acquisition of clinical parameters and also plasma collection for the biochemical and molecular characterization. So at first we isolated microRNA from the plasma of these animals at the different time points, and we assessed by quantitatively and qualitatively on an adjuvant small RNA chips. And we saw that 4 hours prior ALF onset, the global microRNA content starts rising, and the increase is steady until death. And this total microRNA content highly correlates with liver, kidney, and brain clinical parameters. So the next question was to quantify and put a time when the different tissues could contribute to this global increase in microRNA pool in the plasma. So for achieving that, we had to first identify a robust endogenous control which was unaffected by acute liver failure, and of the 3 potential candidates we tested, only one, it was a novel, MIR26A, resulted not to be affected by acute liver failure. So we start testing and quantifying the liver-specific microRNA MIR122, and we showed that the levels of MIR122 started becoming significantly higher and progressively increased from the point of ALF onwards until death. And this the increase in MIR1 to 2 highly correlated with the international normalized ratio. Next, as the kidney failure is a comorbidity in acute liver failure, we quantified the kidney-specific MIR192. And in this case, we saw that there was a significant increase in the levels of this macroRNA 8 hours post-ALF, and it kept going up until death. And this, the time of increase, a significant increase of MIR192 coincided with creatinine levels reaching twice the baseline. And there was a strong correlation with the increase in creatinine and the increase in in MIR192. Last but not least, especially for this audience, we wanted to see as a brain edema and cranial hypertension is a major complications in acute liver failure. We wanted to see quantify brain-specific macroRNAs, MIR124, just to see when and to what extent acute liver failure might affect the brain. And in this case, we saw that MIR124 significantly raised pre-terminally, just soon after ammonia levels became significantly high in the APAP animals, and also this when the levels of intracranial pressure became significant levels compared to the control. And also when the cerebral perfusion pressure dropped below 30 millimeters of mercury. And MIR124 rise correlated strongly with cerebral perfusion pressure. So now if we plot all the tissue-specific macroRNAs together, we can see that the plasma level of these tissue-specific macroRNAs can allow us to visualize a timeline of organ injury in this APAP-induced acute liver failure, with liver-specific MIR122 rising from ALF, followed 8 hours after by kidney-specific 192, and pre-terminally by brain-specific 124. So the next question we want to ask was, what's the biological role of this temporal release in the plasma of the different tissue-specific macroRNAs? So are they molecular signals which the damaged tissue used to communicate to each other, or are they just biomarkers of tissue damage or injury? So in order to achieve that, we isolated from the plasma exosomes, and we purified and extracted both proteins and macroRNAs. And even though the exosomes contained proteins and express nicely markers for the exosome, each represents 70 and CD9A, they were deprived of macroRNAs. So the increase in the macroRNAs in the plasma most probably are associated to proteins in this APAP model. And so if that's the case, most probably they are shed by dying cells. And so we also quantified another damage-associated molecular pattern, genomic DNA, and we saw that the trend in the increase in genomic DNA followed the trend of increase of total macroRNAs, as is shown here. It becomes significantly high at the point of ALF and onwards, and strongly correlates with the global macroRNA increase. So basically, in conclusion, we can say that the studies show that there is an hierarchy in tissue damage, and tissue-specific macroRNAs can serve as tools that can help predict increase in clinical parameters. and with a suitable human population, global and tissue-specific microRNAs should be investigated to determine their prognostic potential as suitable biomarkers in patients with acute liver injury and failure. And I'd like to continue the next.